today I'm excited to be able to introduce you to Dr. Islam, who's going to speak to us about an important paper that he wrote on the mitochondrial calcium uniporter of pulmonary type 2 cells determines severity of acute lung injury, a topic that's very important to so many people. So thank you very much, Dr. Islam, when you're ready to begin.
Thank you very much for giving us the chance to present our data here. So my name is Naeem Islam. I work with Professor Jahar Battacharya in the lung biology lab at Columbia University. And we study mechanisms regulating acute lung injury, or in its severe form, the acute respiratory distress syndrome. Specifically, we study mitochondrial mechanisms and to which extent they regulate acute lung injury. Acute lung injury, or ALI, is a debilitating disease, and it causes pulmonary edema, a condition where liquid floods the alveolar air spaces. And the liquid flooding of the alveolar air spaces shows up as ground glass opacities in the chest X-ray. There are many causes of ALI. However, bacterial infection, specifically gram-negative bacterial infection, is a major cause. And depending on which paper we are reading, the mortality is very high. It's more than 30% of the hospitalized patient. Sadly, therapy is still supportive. There is no specific pharmacological therapy against ALI or ARDS. We are interested in studying mitochondrial mechanisms because reports from our lab and others have shown that in ALI or in ARDS, mitochondria become dysfunctional. And the extent of mitochondrial dysfunction correlates with the severity of the disease. In fact, reports from our lab have shown that when we reinstate mitochondrial function and rescue tissue energetics, animals survive in an LPS model of lung injury. Specifically, what we did is we used LPS, or lipopolysaccharide, to induce acute lung injury in mice. And we gave intranasal instillation of this drug LPS at a lethal dose. And we saw within 4 days, almost 80% of the animals died. But in contrast to that, when we treated the animals with an intranasal dose of bone marrow-derived mesenchymal stem or stromal cells, we saw that these cells were able to transfer viable mitochondria to alveolar epithelial cells. And in doing so, they were able to rescue cellular ATP, reinstate surfactant secretion, and provided protection against LPS inverse lethality. By contrast, when we gave MSCs having dysfunctional mitochondria, those MSCs, even though they transferred mitochondria to the alveolar epithelial cells, they were not protected. So more specifically, we study mitochondrial mechanisms in the alveolar epithelial cells because alveolar epithelium constitutes roughly 90% of the surface area of the lung. And therefore, if there is a disease of the alveolar epithelial region, it can become life-threatening very quickly. And acute lung injury or acute respiratory distress syndrome are diseases of the alveolar epithelial region. This is a schematic diagram of an SNS. This is an alveolus. This is a cuboidal type 2 cell. This is a thin alveolar type 1 cell. The alveolar type 2 cell synthesizes, stores, and secretes pulmonary surfactant. Pulmonary surfactant is a mixture of phospholipids and proteins. When surfactant is released from the type 2 cell, it spreads along the alveolar air-liquid interface. And in doing so, because of its surface active properties, it reduces the surface tension and improves alveolar patency. Surfactant is a prominent safety factor against pulmonary edema or liquid flooding of the alveolar airspace. In conditions such as ALI or ARDS, when surfactant secretion is inhibited or released surfactant is inhibited or blocked or destroyed, the alveoli become unstable and become prone to alveolar edema. And in this project, we wanted to study to what extent mitochondrial dysfunction blocks surfactant secretion. Now, alveolar epithelial mitochondria may regulate a number of homeostatic mechanisms, but in this project, we studied surfactant secretion as a metric for injury severity and mitochondrial function. So this is what the edema would be. And So to study surfactant secretion in intact alveoli of live lung, we established a protocol called lung hyperinflation protocol, which is a stretch stimulus we give by increasing the airway pressure from resting 5 centimeters of water to 15 centimeters of water. And we hold the airway pressure at this elevated level for 15 seconds. And then we bring the airway pressure back to a baseline of 5 centimeters of water. This single transient hyperinflation stimulus causes surfactant secretion from the type 2 cell of a live alveolus. We established this hyperinflation protocol initially in the rat lung. Then we studied this protocol in mouse lung. And lately, we have established this hyperinflation protocol in regards to inducing surfactant secretion in human alveolus. For instance, for our method of studying surfactant secretion is using a confocal or 2-photon confocal microscope. For this, we take out the heart and lung and block, and the excised heart and lung is placed under the microscope, and we inflate the heart lung with a cannula inserted into the trachea, and we hold the airway pressure steady at 5 centimeters of water, and we perfuse the lung with blood, and we hold the pulmonary pressure at 10 centimeters of water and left atrial pressure at 3 centimeters of water. And when we image the alveolar epithelial region with a confocal microscope, we obtain images such as this. In this image, you can see the green fluorescence of mitochondrial matrix targeted GFP being expressed in alveolar type 1 cell, as well as in the cuboidal type 2 cell. And as you can see, the mitochondrial fluorescence is emanating from both type 1 and in type 2 cell. The alveolar airspace doesn't have any fluorescence. Therefore, the alveolar airspace looks dark. So we obtain images such as these. And using this approach, we try to figure out to what extent mitochondrial dysfunction contributes to ALI. As I said, we have now started using our hyperinflation protocol to study surfactant secretion from live human alveoli. For instance, this is an alveolar field of a human lung. The green spots that you see, these are the alveolar type 2 cell of a human alveoli. And if we zoom in of a single type 2 cell, you will see the green spots, which signify the lamellar body, which are the surfactant-containing vesicles that are arranged around the nucleus of a type 2 cell. And the dye is an acidophilic dye, which enters the acidic compartment of the lamellar body. And you can see the green fluorescence of this lamellar body dye that is arranged in the type 2 cell. And when we give this hyperinflation stimulus, we can see two things. One, we can see the green fluorescence of the dye, which is in the lamellar body, progressively decrease over time, indicating that the lamellar body content, that is the surfactant, is being secreted into the alveolar airspace. And we see another dye, which is the red dye, which is FM143, which is in the alveolar airspace, and it is picking up the lipid that is being secreted out of this type 2 cell. And we can see the red fluorescence of the lipid dye increases over time, indicating the surfactant phospholipid is gradually being exocytosed from the lamellar body into the alveolar airspace. So this is how we study surfactant secretion from type 2 cell of live human alveoli.
Amazing.
So the important message here is that the lamellar body localizing dye will show a loss in fluorescence, indicating surfactant secretion. So we have established this hyperinflation protocol for surfactant secretion. Now, calcium is a very important second messenger that regulates surfactant secretion. And since hyperinflation stimulus causes surfactant secretion, we wanted to study whether there is a calcium response as a consequence to the hyperinflation stimulus. We have two movies here. The movie on the left is taken, both movies are from alveolar epithelial region. In this movie in the left, I have stained the alveolar epithelial cytosol with a cytosolic calcium sensing dye fluo4. And in the movie in the right, I have stained the alveolar epithelial mitochondria with a mitochondrial calcium sensing dye, ROD2. Both the movies are divided into 3 segments. In the first segment, you will see the pre-hyperinflation fluorescence. In the second segment, you will see the movie go dark, because in that region, I am hyperinflating the lung, so I'm not taking an image, so image will go dark. And in this third segment, you will see the post-hyperinflation fluorescence change in the alveolar cytosol as well as in the mitochondria. So this movie here, you can see the cytosolic calcium. This is pre-hyperinflation fluorescence. Now I'm hyperinflating. And this is post-hyperinflation. You can see the mean cytosolic fluorescence did not appreciably change. And this is the tracing. In the black trace, we see there is increase in oscillation in the cytosol, but there is no appreciable change in the mean fluorescence in the alveolar epithelial cytosol. As different from that, when we look at the mitochondrial calcium changes following a hyperinflation, this is what we see. Baseline fluorescence, hyperinflation, and post-hyperinflation, you can see an appreciable change in fluorescence in the alveolar epithelial mitochondria. And this is the tracing. In the red tracing, we see the baseline fluorescence. And following hyperinflation, we see mean mitochondrial calcium increase in the alveolar epithelial mitochondria. So this is a schematic diagram that shows the calcium handling machinery in a typical type 2 cell. When we hyperinflate the lung and provide a stretch stimulus, calcium is released from the store, which is the endoplasmic reticulum. Calcium that is released is pumped back into the ER. Some of it is pumped back outside of the cell from using the plasma membrane calcium ATPase on the membrane. But there is this mitochondria, which is very close to the ER. In fact, mitochondria and ER are in contact with each other with tethering proteins. And on the mitochondrial inner membrane, we have the MCU, which is the red channel here. And we have the ancillary proteins adjacent to the MCU, which may regulate the MCU channel activity. Some of the calcium that is released from the ER is taken up into the mitochondrial matrix through the MCU. The entry of calcium into the mitochondrial matrix through the MCU is along this proton gradient. Mitochondrial matrix is a highly negatively charged region, and calcium entry is along this electronegative charge. So what we are suggesting is that lung hyperinflation causes mitochondrial calcium entry. And we know that mitochondrial calcium entry into the matrix is important because there are a number of matrix-bound dehydrogenase enzymes which require calcium as a cofactor to initiate ATP production. And this ATP is a surfactagogue, which releases surfactant secretion, causes surfactant secretion, which promotes oxygenation and maintains alveolar patency and provides a safety factor against edema. Now, all this calcium entry, ATP production, surfactant release, everything depends on this MCU channel because this is the channel that regulates calcium entry into the mitochondrial matrix. So we wanted to answer whether MCU dysfunction contribute to ALI. To study MCU dysfunction, we generated mice lacking the MCU or mitochondrial calcium uniporter channel in the alveolar type 2 cell. To do so, we bred the MCU floxed mice with SPC Cre mice, where the Cre recombinase is targeted with an SPC promoter. And the offspring don't have the MCU in the type 2 cell. The immunoblot here shows that MCU flox mice has the MCU in type 2 mitochondrial fraction. As different from that, in mice where we activated the Cre, the MCU in type 2 mitochondrial fraction was lost. Since MCU is lost in these mice, we wanted to figure out if there is calcium buffering, or will these mitochondria allow calcium to enter the matrix? And we use the hyperinflation stimulus. In the flox mice, we see mitochondrial calcium entry into the matrix. As different from that, in the type 2 cell specific MCU knockout mice, the hyperinflation-induced mitochondrial calcium entry was blocked. Since mitochondrial calcium entry is blocked in the MCU knockout mice, will it also inhibit surfactant secretion? For this, again, we stain the type 2 cell lamellar body with this lamellar body localizing dye. This is a single lamellar type 2 cell in an intact alveolus of a live lung. And you can appreciate in the flox mice, hyperinflation caused loss of fluorescence from the lamellar body, indicating surfactant secretion. As different from that, in the type 2 specific MCU knockout mice, the loss of fluorescence in lamellar body dye was abrogated, indicating that hyperinflation-induced surfactant secretion was blocked in cells where MCU is lacking. So this is what we think, that lung hyperinflation, which causes mitochondrial calcium entry, which induces ATP production, leading to surfactant secretion. And when we block calcium entry through the MCU channel, surfactant secretion is inhibited. To study alveolar stress responses, we again established the LPS model for ALI or ARDS. For this, we intranasally instill LPS at a non-lethal dose of 1 milligram per kilogram dose. And we saw that at this dose, there is robust inflammation. For instance, in this image here, we have stained the alveolar epithelial cytosol with a cytosolic calcium sensing dye fluo4. And these two images were taken by my colleague, Christine Westphalen. And in the left image, you can see the fluo4 stained alveolar region. And we have also stained CD11B to see if there is any extravasate leukocyte in the alveolar region. In control lung, where there is no disease, the alveolar region is free of leukocyte. As different from that, in ALI or LPS-treated lung, we can see numerous leukocytes crawling around in the alveolar airspace. You can see all these leukocytes are stained with CD11B antibody, and they are crawling around in the alveolar region. So this dose of LPS at 1 milligram per kilogram caused robust inflammation, but no death. And we wanted to find out if, since we have already presented in our previous paper that LPS blocks surfactant secretion, and we wanted to reconfirm that, again, LPS will block surfactant secretion. You can see in PBS-treated lung, lamellar body fluorescence goes down with hyperinflation, indicating surfactant secretion. As different from that, in LPS-treated lung, lamellar body fluorescence do not go down, indicating, again, that LPS blocks surfactant secretion. In addition to causing inflammation marked by leukocyte extravasation and loss of surfactant secretion, at this dose, LPS was able to increase extravascular lung water, a marker for pulmonary edema. So we are using a dose of LPS which causes inflammation, but not death. So here's our hypothesis. LPS is causing inflammation, blocking surfactant secretion, and causes alveoli to become more prone to alveolar edema. Since surfactant secretion is blocked in MCU knockout mice, and surfactant secretion is also blocked in LPS-treated mice, we wanted to figure out if LPS has any effect on the MCU of type 2 cell. For this, we gave LPS, again, at the non-lethal dose, and we took out lung and extracted the type 2 cell mitochondrial fraction at different time points after LPS instillation. And we saw that at 24 hours, the time point where we see edema, the time point where we see abrogation of surfactant secretion, the time point where we see extravasation of leukocyte, at that time point, there is a significant decrease in MCU protein content in the alveolar type 2 cell mitochondrial fraction. However, you can see that the MCU protein content gradually increased, and by day 5, the protein content was almost back to the pre-LPS concentration. As different from the non-lethal dose, when we give intranasal instillation of LPS at a lethal dose, a dose that kills 80% of the animal, we see, again, at 24 hours, a significant drop in MCU content in the type 2 cell mitochondrial fraction. But as different from the non-lethal dose, at this dose, the MCU content do not recover. Interestingly, when we plot the MCU level, we see that in non-lethal dose, MCU content recovers and comes back to baseline by day 5. As different from that, in the lethal dose, MCU content do not recover. This is the body weight, which is a marker for animal health and metabolic activity. We see the body weight also goes down on day 1, and body weight changes mirrors MCU content in that with the non-lethal dose, body weight the animal starts to gain back weight, and by day 5, at a time point where MCU has come back to baseline level, body weight also comes back to baseline level. As different from that, when we gave LD80 dose, where animals don't survive, or 80% animals don't survive, we see body weight of the animals don't recover.
Mm.
So in the next set of experiments, we gave LD20 dose, a dose that kills 20% of the animal in the control mice, and we see 20% of the MCU flox mice dies at this dose. As different from that, in the type 2 specific MCU knockout mice, there was a significant increase in lethality, indicating that loss of MCU in the type 2 cell exacerbates MCU LPS-induced lethality in these mice. Almost 90% of the mice died at this dose, which killed only 20% of the flox mice. So we wanted to find out whether LPS-induced loss of MCU abrogates mitochondrial buffering. Again, I have 2 movies here. In both movies, we have stained the alveolar epithelial cytosol, alveolar epithelial mitochondria with a calcium-sensing dye ROD2. The movie in the left is from control lung, and the movie in the right is from LPS-treated lung at 24 hours post-LPS installation. Again, the movies are divided into 3 phases. This is the PBS or control lung baseline, hyperinflation. Following hyperinflation, there is a robust increase in fluorescence indicating epithelial mitochondria are buffering calcium. As different from that, in the LPS-treated lung, post-hyperinflation, there is no change in fluorescence, indicating mitochondria are not able to buffer, because at that time point, MCU in the type 2 cell is lost. And here's the group data. PBS lung, we see buffering. As different from that, in the LPS lung, we see hyperinflation-induced buffering is lost. To define why MCU is being lost, we concentrated on reactive oxygen species, because reactive oxygen species is a very prominent signaling molecule that can regulate a number of cellular function, including proteases. And we confirmed that following LPS installation at 24 hours level, time point where we see robust loss of MCU, at that time point, there is a lot of reactive oxygen species. For this, we stained the alveolar epithelial mitochondria with a mitochondrial matrix targeted Rho-GFP protein. This is a sensor for reactive oxygen species. High levels of reactive oxygen species oxidizes this protein and reduces fluorescence. As you can see, compared to control lung, in the LPS-treated lung, the fluorescence has gone down because it has been oxidized. To further study the role of reactive oxygen species, we created a transgenic mice which overexpresses human catalase in the mitochondrial matrix of alveolar type 2 cell. For this, we bred the alveolar mitochondrial catalase flox mice with the SPC Cree mice, and we overexpressed catalase to mop up reactive oxygen species in the type 2 cell mitochondrial matrix. And using these mice, we saw that LPS caused a robust increase in reactive oxygen species in AMCAT flox mice. As different from that, when we have AMCAT overexpression in the type 2 cell, the increase in reactive oxygen species was blocked. And in these mice, we immunoblotted for MCU at 24 hours after LPS installation. And we can see that in wild-type mice, at 24 hours after LPS installation, LPS causes robust loss of MCU. In AMCAT flox mice, there is significant loss of MCU. As different from that, in 82 specific AMCAT overexpressing mice, there was the LPS-induced loss of MCU was abrogated, indicating when we remove reactive oxygen species by catalase, the loss of MCU is blocked. Mm-hmm. To define whether LPS is causing a deregulation of mitochondrial arrangement in the alveolar type 2 cell, we obtained a dendra mice which expresses mitochondrial matrix targeted fluorescent protein GFP here, dendra fluorescent protein. This is a zoomed image of a single type 2 cell. You can see the mitochondria are arranged around the nucleus of a type 2 cell. And the mitochondria, and in the red channel, you can see LTR, which is the lamellar body localizing dye, which is staining the surfactant-containing lamellar bodies in the type 2 cell. And in the merge channel, you can see in the PBS or in control lung, the surfactant-containing lamellar bodies are in closed proximity to the mitochondria of the type 2 cell. And you can see the mitochondria are arranged in bipolar distribution in the typical type 2 cell. But as different from that, in the LPS-treated lung, again, after 24 hours, we see that the mitochondrial distribution has changed in that the mitochondrial proximity to the lamellar body has decreased, and the mitochondria has lost its polar distribution. There is deregulation of mitochondrial arrangement in the LPS-treated type 2 cell. Why is this dearrangement we are seeing here? For this, we concentrated on a protein called dynamine-related protein, or DRIP1. DRIP1 is a large cytosolic GTPase, which can translocate to mitochondria due to a number of physiological stimuli. And we wanted to find out whether DRIP1 is translocating to type 2 cell mitochondria following LPS. For this, we immunoblotted for DRIP1 in the type 2 cell mitochondrial fraction. And we see that at 24 hours following LPS installation, there is a robust increase in DRIP1 in the type 2 cell mitochondria. One such stimuli that causes DRIP1 to phosphorylate and translocate to mitochondria is reactive oxygen species, which phosphorylates DRIP1 at serine 616, causing DRIP1 to translocate to mitochondria. And we immunoblotted for phosphoserine 616 DRIP1, and we see that in mitochondrial fraction of type 2 cell, LPS caused a robust increase in phosphoserine 616 DRIP1, indicating that in our model, reactive oxygen species is phosphorylating DRIP1 at serine 616, causing it to translocate to mitochondria. And DRIP1 is a mitochondrial fission protein, which causes mitochondrial fission. And to further study DRIP1 effect in MCU, we generated DRIP1 knockout mice by breeding DRIP1 flox mice with SPC3 mice. And we see that following LPS in the DRIP1 flox mice, there is loss of MCU. But in the type 2 specific DRIP1 knockout mice, the loss of MCU that we see with LPS in flox mice, that loss was abrogated, indicating when we knock out DRIP1, loss of MCU is also blocked. And in the last set of experiments, we wanted to study whether we can use MCU as a therapy for ALI or ARDS. For this, in the type 2 specific MCU knockout mice, we reintroduced the full-length MCU, which is what we call add-back experiment. We added back MCU in the type 2 specific MCU knockout mice. And when we added back MCU, we saw that the hyperinflation-induced surfactant secretion, which was blocked in the MCU knockout mice, that was rescued by the add-back of MCU in the MCU knockout mice. Also, we measured pulmonary edema, which is our metrics for measuring pulmonary edema is measuring blood-free lung water. We see that following LPS, there is an increase in pulmonary edema. And the increase in pulmonary edema that we see in the MCU flox mice was blocked in mice where we added back MCU, indicating add back of MCU reinstates surfactant secretion and prevents LPS-induced edema. And this is, we started this experiment and with LPS, given LPS at a dose of LD20, and we saw that whereas only 20% of the flox mice died, as different from that, in type 2 specific MCU knockout mice, there was a catastrophic increase in lethality in that 90% of these mice died. However, when we add back MCU to these mice, we see a significant increase in protection in that add back of MCU in type 2 cell rescued surfactant secretion, prevented edema, and increased animal survival. So this is our summary. We think LPS induces mitochondrial reactive oxygen species, which phosphorylates DRIP1, causing DRIP1 to translocate to mitochondria, causing mitochondrial deregulation, leading to MCU loss, which causes loss of surfactant secretion, which increases alveolar increases severity of injury, makes alveoli more prone to edema. However, when we inhibit mitochondrial reactive oxygen species with mice overexpressing catalase in the type 2 cell mitochondria, or when we inhibit DRIP1 with mice lacking DRIP1 in alveolar type 2 cell, we reverse all LTS-induced effect in that there is MCU gain, there is rescue of surfactant secretion, and lung injury is ameliorated. So this is our publication. I want to thank my mentor, Jahar Battacharya, for his guidance throughout this project and for his support. And I want to thank all my lab members for helping me with this project. And I want to thank this organ repository Live on New York for providing us with human lung, and Dr. Donna Farber for executing the MTA with Live on New York and providing us with the human lung. And these are our funding sources. And again, Mary, thank you for giving us the chance to present our data in this conference. Thank you.
You are a wonderful teacher and scientist. I am not a basic scientist, but you were able to explain it in a way that I could see all the clinical implications for your work and the imaging implications. I think about COVID-19 and how this has so much application for treating patients because they had so much surfactant deficiency, and we prone positioned them to try to compensate for the surfactant deficiency to increase their lung aeration. I have one question. Fibrosis of the lung is very important to me. And I believe I've studied how collapse in duration, so the lack of surfactant leads to atelectasis, and when that's persistent, fibrosis development. And I always thought that if we had given surfactant earlier on, we might never get to the point where we needed medications to combat collagen. I wonder if you think about implications of the treatment that you described might help prevent fibrosis from occurring.
Thank you for asking this question. So the role of surfactant in pulmonary fibrosis is probably a little understudied at this moment. However, the role of type 2 cell in pulmonary fibrosis has been studied a lot. And one of the major causative reasons that lead to pulmonary fibrosis is assumed to be the type 2 cell senescence. So, what, in terms of type 2 cell dysfunction and fibrosis, what the role of surfactant could be that with age, there may be sporadic loss of surfactant secretion. In fact, we have studied surfactant secretion in human lung, and we see that in human lung, there are regions of high collagen deposition. And in regions of high collagen deposition, we see hyperinflation in the surfactant secretion is decreased. And we see with age, these regions of high collagen deposition increases. There is disparity in collagen deposition in all age group, but with age, generally there is thicker subpleural collagen deposition. And in regions, alveolar regions adjacent to thicker subpleural collagen deposition, we see loss of hyperinflation in the surfactant secretion. So while we probably do not have the answer yet, but our working hypothesis at this point is that persistent loss of surfactant secretion would cause a drop in interstitial pressure, which would increase microvascular filtration and probably make these regions more prone to profibrotic events. And if that happens for over a period of time, those stimuli put together may make these regions more profibrotic, leading to what generation of what we think are interstitial lung abnormalities, those foci which are correlated to interstitial lung diseases. So this is, I suppose, a more of a hand-waving answer, but this is our working hypothesis, I suppose, at this moment.
Thank you so much. Dr. Bhattacharya, would you like to say something?
Well, it's a very interesting question. And I agree with Naeem, it's really not known. So what the connection is between, exact connection is between mitochondria, especially type 2 cells, and the fibrotic process. To my knowledge, theories of pulmonary fibrosis, as we understand them from other people's work, is that you got to have some kind of a monocyte infiltration from the blood, from bone marrow derived monocytes. And these interact with fibroblasts to cause collagen synthesis. And this other one is related to telomere shortening.
Mm-hmm.
So if you have telomere shortening, somehow that, I'm not sure how, but that somehow makes, I guess it leads to oxidant production and
Probably.
fibrosis. Here we have a situation where the mitochondria are becoming dysfunctional. And there is a theory out there that if you, this calcium uniporter may have a role in the fibrotic process, but it's very vague. It's not clear to me how to connect the mitochondria. I'm saying if the mitochondria become overactive due to some, so another theory of fibrosis is that there is a infection in the epithelium, which then causes release of TGF-beta, right, through the IL-1 alpha V beta 6 mechanism. This is the Dean Shepherd theory of fibrosis. Now, if that's the case, then is it possible that the oxidants released from mitochondria by overactivity of the mitochondrial calcium uniporter, could those impact fibroblast function and lead to TGF-beta secretion, and therefore from the epithelium, I guess, or from the intestine, activate TGF-beta in the connective tissue and cause fibroblast activation and therefore collagen production. I mean, could there be a pathway? But, you know, again, these are all hypotheses waiting to be tested. Currently, models of fibrosis tend to be such difficult models in mice. Mm-hmm. If you use bleomycin, I mean, really, gosh, it's like a blunderbuss approach. I don't think those mechanisms. I always find it difficult to understand human fibrosis on the basis of bleomycin.
Because it's reversible and human fibrosis is not, right?
Yeah. It's such a subtle development, which then becomes very strong. So it may be senescence, as Naeem points out, but how do you connect senescence to the fibroblast? That's the question, right? Where's the senescence? Is it type 2? Is it fibroblast? So there's far too many questions here.
So we'll have plenty of work for a long time, right? Because there's so many questions.
I think so.
I love every interaction I get to have with you, and it's so nice to get to meet Dr. Islam today as well.